Hello, and welcome to our presentation on rabies virus transcription and replication. My name is Loudon Hewlett, and Bryce Schmidt will be the presenter in the second half of the video. The outline of the presentation will follow the molecular biology associated with the rabies virus. Virus particles are responsible for transcription and replication, and studies that have been done to understand transcription and replication of the rabies virus, and potential future studies that can be done to understand more. We will first start with the pathogenesis. Rabies is a zoonotic disease spread by a variety of mammals to humans, examples being rodents and domesticated animals such as dogs. The virus is shed from saliva, from the animal's salivary glands. The virus can survive for multiple days within the animal's saliva, but once exposed to direct sunlight, the virus would only survive for at most an hour, meaning that the virus must be spread quickly and usually through an animal bite. Once the infected saliva enters the human body, the human tissue cells are the first to be infected. However, over varying incubation periods that could be soon before 20 days or longer than 90 days, the virus invades the nervous system and makes its way to the brain. At this point, once inside the brain, the lethality of the disease is nearly 100%. However, the invasion of the CNS by rabies will be covered in another lecture by another group. Now to get started. Now to break down the rabies virus. Rabies lyssavirus is the scientific name given to the rabies virus. The virus is a single-stranded, negative-sense, non-segmented, enveloped RNA virus. The virus takes a bullet-shaped form with its genome consisting of around 12,000 nucleotides. The genome is composed of 5 different viral genes, labeled as N, P, M, G, and L, encoded into different regions of the RNA. The genes are all composed of internal coding regions flanked by non-coding regions. These regions are bordered by initiation and termination sequences in the genome. The genes are also separated by non-transcribed enterogenic regions, which vary in length between genes and help with the regulation of transcription and replication. A fully formed genome is wrapped and surrounded by the nucleoprotein labeled as N. This protein helps form the RNA N complex for transcription and replication of viral RDRP, labeled as L, and its cofactor phosphoprotein, labeled as P.
Hello class, this is Bryce Schmidt, and I will continue with an explanation of transcription. Viral proteins N, P, and L will form complexes that act as RNA-dependent RNA polymerases to transcribe the genome of the virus. The virus is transcribed beginning with the 3' end leader. The leader initiates transcription of the genome and is non-capped and non-polyadenylated. The other 5 viral genes, however, will be capped and polyadenylated during transcription. The genome will then be transcribed in a start-stop method, with the complex dissociating at each stop terminal and reinitiating poorly at the next initiation site. This forms a concentration gradient described in the next slide. Poly-A tails on the viral gene will be synthesized due to slippage at the termination points on the end of the genes. The figures on this slide illustrate further the process of transcription of the viral genome. The first figure, figure A, showing the viral cycle. Rabies virus replicates in the cytoplasm of the host cell. Several phases can be observed. Receptor binding, endocytosis, followed by membrane fusion and nucleocapsid release. Then transcription, replication, and finally virus assembly and progeny virion budding. Within the cytoplasm, the virus will form inclusion bodies, known as Negri bodies, where both transcription and replication will occur. As well, Figure B demonstrates the concentration gradient of viral genes during transcription. Viral mRNAs are produced during transcription naked, which is free of the N protein. The leader RNA is neither polyadenylated nor capped, whereas viral mRNAs are polyadenylated at the 3' end and capped at the 5' extremity of the L subunit. Reinitiation of the transcription does not always occur, generating gradient amounts of the transcript. Leader RNA having the most product, and mRNA L having the least abundance. So viral negative sense and positive sense RNA are produced during replication and are always associated with NP and L. So examples of experiments used to gather this information are the following. The compact bullet-shaped virion and interactions with the M protein were determined by cryo-electron microscopy. The transcriptional control for a negative non-segmented RNA virus is due to the control of RNA-dependent RNA polymerases. Genome sequences using molecular cloning to indicate genes and non-coding regions. Some potential next steps include new techniques such as fluorescent tracking of rabies virus can help illustrate the specific mechanisms promoting the viral pathogenicity, as well as understanding processes for non-segmented transcriptional initiation and termination. Here are references, and thank you for learning with us.